Hello everyone, I'm here very glad to show you our recent work, and the title is Targeting Keystone Species Helps Restore the Dysbiosis of butyrate-producing bacteria in non-alcoholic fatty liver disease. So non-alcoholic fatty liver disease, which we also call it NAFLD, is a complex multifactorial disease whose pathogenesis remains unclear, and the advanced form of NAFLD with hepatic inflammation is nonalcoholic steatohepatitis, that is NASH. The exact pathogenesis of NAFLD is still unclear, and the theory of multi-hit is widely accepted. Many studies have shown that dysbiosis in the gut microbiota affects the initiation and development of NAFLD. Many microbial intervention therapies targeting the gut microbiota are being considered to treat NAFLD. However, these microbial interventions have not achieved satisfactory effects, which may be partly explained by the structural complexity of the ecosystem we have in our gut. And therefore, further studies of the dynamic change in the microbiome during disease development are essential for the identification of microbial targets and the precise treatment of NAFLD. Research have shown that keystone species represent excellent candidate targets for gut microbiome-based interventions, as they are defined as the species required for the integrity and stability of the ecological system. Many different methods have been used for microecosystem modeling to help identify keystone species in microbiome, but they have shortcomings in applications. Causality methods, by contrast, it benefits from the prior knowledges and is more useful to identify real interactions. And by using it, we can capture dynamic process of the impact of keystone species on the entire community. As such, in this study, causal algorithm integrated with ecological theory was used to model the microecosystem. And at the same time, we also applied dynamic intervention simulation to capture the dynamic changes of the network during intervention and purposefully evaluate the change from disease to normal states. For study design, here we collected stool samples from 22 patients with NASH and 16 healthy individuals for gene sequencing. Causal algorithm was used to model the microecosystem, and keystone species were then identified through the dynamic intervention simulation. This strategy was also applied to a set of 25 obesity patients and an external validation cohort for further comparison. So in our result, we found that the gut microbiome in NAFLD seemed to be decreased abundances in Lachnospiraceae and Ruminococcaceae. As the major butyrate-producing bacteria in the intestine, these bacteria may play important roles in suppressing intestinal inflammation and can consequently suppress the pathogenesis of NASH. Then, by using causal inference, we constructed microbial interaction networks of the normal, obese, and NASH subjects, and we found that the gut microbiome of these three groups exhibited distinct patterns of microbial interaction. And then, by dynamic intervention simulation, we identified 8 keystone species of NASH, including Porphyromonas, loveana, Alistipes, indistinctus, and Dialister, pneumosintes. Targeting these keystone species, the microbiome of the patients can be maximally restored toward normal status, and in particular, the abundance of species in Lachnospiraceae and Ruminococcaceae increased after intervention. Though these are the microbial interaction networks of NASH, the yellow lightning represents the targeted species, and all the affected species are marked with circles. Through these networks, we can directly see that the intervention of keystone species could restore the abundances of many gut microbes, thereby promoting the reconstruction of a normal intestinal microbiome. At the functional level, the keystone species could regulate intestinal amino acid metabolism and acid-base environment so that to promote the growth of the butyrate-producing species. Here are the 3 take-home messages. That's all. Thank you very much. And if you have any questions or you are interested in our research, please feel free to contact us through email.